Hello children, I believe every one of you who is watching this video is doing good, and thank you for showing so much love for all the biology videos and also the other videos out there. I'm really glad that I get a chance to teach you guys. So this is your Jasmine Ma'am, and I'm here with the most important questions from the chapter Principles of Inheritance and Variation. One very important thing before I go ahead, and that is for some children this chapter is there, for some children this chapter has not been completed fully. In that particular case, do understand, I'm giving you the list of questions. Whichever topic has not been taught to you, you can just, what do you say, unsee it, okay? Do not go ahead, and if you do not want to invest too much time into it, you can just ignore that particular question. Okay, cool. Now let me show you the questions, and also one more very important thing. So for every other chapter, sexual reproduction in flowering plants, human reproduction, as well as reproductive health, okay, I have done the one-shot videos. I think reproductive health video is going to be out today or probably by tomorrow, but there will be one-shot videos that are available. For principles of inheritance and variation, I have not done the one-shot. I came in the live yesterday in the second marathon, and I had discussed all the important questions that I've been going to talk about over here. But still, if you feel that, ma'am, we want the answers in general, we want to just read through it. So since I have not done the one-shot yet, I thought I should be helping you with the answers. So here in this particular thing, you are going to find the answers as well. Okay, so do not be worried. You do not need to be, what do you say, searching for the answers and things like that. I'll tell you the way where you can download the PDF. Okay, let's go ahead. So usually in boards, you get 1 2-marker question from this chapter and 2 5-marker questions from this chapter. But now since it is not board, it is just your FA examination, it can be 2, 3, 5-marker, anything that could be coming from this chapter. But the important topics remains to be the same only. First question says, in Drosophila, genes for yellow body and white eye show lesser recombination, and white eye and miniature wing show very high recombination. Explain why. So basically you have been asked about a concept which is known as linkage and recombination. Now that can come in different format. Basically they can ask you what is the difference between linkage and recombination, or they can give you a question like this where they're giving a scenario that why is this happening, that basically over here, Baba, the question is like this. Okay, that is genes for yellow body and white eye. Suppose this is a gene for yellow body and this is a gene for white eye. Okay, they show lesser recombination, but white eye and miniature wing. So white eye was here, yellow body gene was here. Okay, and now you have been asked about miniature wing gene. Suppose miniature wing gene is over here. How do I come to know that? Because they have told that in the, what do you say, question. How, ma'am? They have not said that this is located like this. They have said one very important thing. Genes for yellow body and white eye show lesser recombination. So yellow body and white eye, when I say that they are showing lesser recombination, that means that they are very close to each other. How do I get to understand that? So usually what happens is, Baba, understand, the chromosomes, when they lie close to each other, no, they are going to have a process that is known as crossing over. They cross over their parts. Suppose I cut from here, this portion comes there, this portion goes there. In that case, what will I get? Let's make a new chromosome. This time, since the portion went there, the white eye, yellow body gene is on the other chromosome, and the miniature wing gene is on this chromosome. Am I not making a new combination? Yes, I am making a new combination. So over here, I still see that both of these genes are together. Why? Because they are closely linked to each other. That is why they are together always. You get it? But this guy is far away. That is why they show higher recombination. Okay, that is why you see higher recombination. New combination is being formed. In the case of this and this, they are not together anymore. That is the basic logic. And I'll tell you easy way to remember this. You and your best friend. Suppose you guys are very close. There are very less chances for anyone new to come. Okay, too much linkage, less recombination. But you and your friend, not too much close, someone else has come, someone else also comes. What happens? Basically, a new combination is being formed because you were not too close. So you can remember like that. I was just trying to help you with a little explanation over here. I'm not going to explain everything. Do not be worried. So this is something like this, and basically on the basis of the same concept, you can get questions like linkage and recombination. What is the difference between that? Or they can ask you recombinants obtained in the Mendelian and the Morgan experiments are different, yet the process involved are different. Comment. So recombinants were obtained in both the Mendel's experiment and Morgan's experiment. Morgan's experiment was on this anyway, yet the processes involved are different. Comment. So you have to talk that in Mendel's experiment, why did you get recombinations or recombinants out there? Because there was law of independent assortment which was working. Now if you do not understand what I am saying, do not be worried. Okay, do not be worried. The reason I am telling you, I told you before also, if you do not know something and that creates anxiousness in you, understand that at least I know that this is a problem and I have to solve it. Now you know that you are going to solve it. How? Probably you can see the answer directly and try to understand, or probably watch my video in the marathon, or if you feel that no, this teacher is not really good, I want to check it from other teacher, you can go that. You can use ChatGPT. Whatever works for you, you can do. But at least you know the type of questions that could be coming in, no? So be affirmative, be good about it, be feeling good about it, don't be sad, okay? Fine. So these type of questions could be coming from the same topic. Next topic is test cross. They can directly ask you what is a test cross, what is the importance of it. Then haplodiploid sex determination or any form of sex determination can be asked for that matter. But I've just given you this particular topic such that you understand how the type of questions could be framed. What is haplodiploid sex determination? Mention an animal which exhibits it. So it is honeybee and you have to talk about it. They can ask you about female heterogamety, then you talk about birds. They can ask you about male heterogamety. What do you mean by female heterogamety? Females produce two type of gametes. Okay, male heterogamety means male produce two type of gametes. For example, in us human beings, males produce X and Y gametes, okay, but in us females it's just X and X. So here, since males are producing two different type of gametes, we say it is heterogamety, male heterogamety. In female heterogamety, the example is bird. Anyways, so these type of questions could be coming in. Next, the difference between incomplete dominance and codominance. Where do you see incomplete dominance? Snapdragon flower. Where do you see codominance in your NCERT? You have to know that is basically your AB blood group. Now, difference between pleiotropy and polygyny, important. Okay, a woman with blood group O married a woman with blood group AB and show the possible blood group. This type of questions could be coming in. So while you go ahead with codominance, while you study about this, that means you're going to study about the blood group. While you study about the blood group, try solving this question also. Check whether you are able to solve this question also. Listen, that's why I'm telling you, no one on this earth can tell that this is a fixed question. This question will only come. If someone says that, then they are basically doing a very wrong thing. And I am not here to blindfold you and say that these questions are only going to come. I am giving you the concept, the most important concepts from where questions usually come. Okay, so these are the important concepts that my dear children have to practice. Then define point mutation. Where do you see sickle cell anemia as an example for that? Talk about it. Okay, identify the symbols in the pedigree analysis. This symbol where you have two lines, that means too close, that means they are mating between relatives. This is happening over here. And yes, these type of questions could be coming here. And this is actually based on this question where it say that a pea plant with purple flowers were crossed with pea plant with white flowers. The progeny were all purple. If only one character is seen in the first generation, that means the purple color is dominant out there. So law of dominance. On selfing, these plants produce 482 plants with purple flowers and 162 plants with white flowers. 3:1 ratio. 3 of them tall, 1 of them dwarf. So over here, 462 basically is the higher number, that is purple. 3 of them purple, and at the end 1 of them white. If at all you come by the ratio, you'll get the same thing. So what does it show? Mendel's law of inheritance. First is law of dominance, that's showed in first generation, and then you have the law of segregation that has been seen. So you have to explain that as I said, if you feel that ma'am, I do not know how to frame my answer, I have shown you a way, you can follow that. Cool? That means that is right over here. You need not need to worry about anything that is there. Cool. Yeah. Now talking about the 5-marker question, mostly the schematic representation based question could be coming in. They can ask you one gene inheritance, show the inheritance pattern. Basically, tall plant, dwarf plant, crossing them and things like that. While that question is being asked, please understand, you have to write, parents, gametes, F1 generation and things like that. Phenotypic ratio, genotypic ratio, know about all these small steps that you need to keep in mind. Okay, so that is one gene inheritance. Now 2 gene inheritance, that is dihybrid cross, can be asked. They can ask you in a different way. Show the cross between plants that is different in seed color and seed shape. Since 2 characters been considered, please understand it's a dihybrid cross. Okay, incomplete dominance in snapdragon, that can be asked to you to make the entire theme schematic representation. Again, the phenotypic and genotypic ratio of every generation, you should be knowing it. Okay, yes. Now that type of question has higher chances of coming in a 5 marker. So be prepared for this. Now observe the representation of a dihybrid cross and answer the questions that follows. So this is a dihybrid cross experiment thing only. You have been asked write the phenotypic ratio. So 9:3:3:1 is a phenotypic ratio. Identify the homozygous dominant. Homozygous dominant means the alleles have to be similar. That is when you call it homozygous. And dominant means dominant character has to be taken. So round and yellow is the dominant one. So similar type of questions are being asked over here. Okay, identify the recessive genotype, number of parental type of progenies, as in progenies which looks just like their parents. Okay, a number of recombinant type of progenies. You have to talk about it. Cool. And I'll give you a trick. If 9:3:3:1 is there, 9 of them will show the dominant character, round and yellow. 1 of them are going to show the recessive character, wrinkled and green. These 2 characters are parental characters. So I can say 9 plus 1, 10 of them are going to be like parents. These 6 of them are going to be something that is going to show non-parental or recombinant new combinations out there. Anyways, just want to tell you. Now, disease related questions could also be coming in. While I kept this question, I need you to understand that this question is a sum of lots of diseases. So while you try to solve this question, basically you have to go ahead with each of the diseases separately. Sex-linked recessive disorder occurring in only 8% of males and only 0.1% of females, leading to defect in the cone of the eye, color blindness. Know everything about color blindness, which is say any type of questions could be coming from color blindness. Now, sex-linked recessive disorder that is leading to leading to a problem in clotting of blood, hemophilia. There are only 2 sex-linked disorders only in your NCERT, color blindness and hemophilia. Next, inborn error of metabolism, accumulation of phenylalanine, that is what you say phenylketonuria, PKU is the disease out there. Autosomal linked disease where substitution is happening, substitution of amino acid, sickle cell anemia. Or autosomal linked disease where basically reduced rate of synthesis, that means quantity is being affected. Quantity of hemoglobin is being affected, that means you're talking about what quantitative disorder? Thalassemia. So basically, as I said, this type of questions are there. It had come before, but I am trying to tell you that whatever disease that I'm talking over here, you need to go in depth, a little bit depth about that. You can get questions on the basis of that. And my dear children, yesterday I was here until I think 9:50. Okay, I think I was there until 9:50 or something 10 o'clock, something like that. I had made sure that at least I tried to make you understand about the concepts which are there for principles of inheritance and variation and also human reproduction. Okay, also human reproduction, but principles of inheritance, as I said, there is no one-shot video that I have provided, which is why this particular lecture can be useful for you. The part 2 of FA1 marathon, it was a live. Make sure you go check it out once. It could be helpful for you, even though there were so many people who were watching the video at that time, because I feel the channel discovery would be taking time. You might not be knowing too much about the channel. Okay, so usually the peak comes towards the examination, but still I would love to tell you, try it out. If it works for you, then what's the bad deal about it, right? And by the way, every one of you who is watching this video and have not hit on the subscribe button, make sure you do so, because this is where you'll find me and I'm going to help you out with biology. Okay, now identify the genetic disorders given below. This is actually previous year FA question. Sterile females, chromosomal complement 45 with XO. There is one X that is not there. That means it's Turner syndrome. Short-statured, furrowed tongue, characteristics palm crease, Down syndrome. Sterile males, gynecomastia has been formation of the breast. Karyotype 47XXY. It is going to be Klinefelter syndrome. So again, while you go ahead and probably look at these answers over here, I have spoke more about it such that you get an idea about all the disease. Yes, and I have taken the help of AI to form it, but still it is definitely going to help you out. And I'll request you, if you have got time post the examination, make sure you watch this one-shot video. Definitely something that is going to help you out. I got so much love in this particular video. I'm expecting the same to happen for all the others. This has not been released yet. So yes, definitely it could be helpful for you. Cool. So yeah, and where do you find the PDF? That is the important question. So Baba, if at all you see under every single video that we put, just give me a second. I have to close this up. Under every single video that we put over here, you have got, if you just roll down a bit, you will be seeing the WhatsApp channel. So we have a lot of children out there. Those are their seniors who joined with us. But I request you guys to come in. Okay, this is where you will be getting your PDF for now. This is very important. And I'll make sure that when the video is being published, after some time, you will be getting the PDF of the same as well, along with the answers today itself. Okay, so I might have a class. That's why I said I'm not sure about the timing and all, but yes, that would be done. And the second part of the story is at the top over here, you could see something like this. Join the free ACE KCET series. This is actually an all-in-one page that is from Vedantu, and over here you just have to go with buy now. It's actually for 0 rupees. You do not have to do any payment, so do not be worried. Basically what happens over here is that once you go ahead with buy now option, you have to fill in your mobile number and stuff like that, but post that you will be able to access each and every video that we have done of academics here in this particular portal itself. So in YouTube it might be sounding to be very confused because counseling videos are also there, but there only subject wise things are there. So in each subject, whatever videos have been done, it will be there. This is the all-in-one page. Make sure you check that out, and as I said, that is not going to harm you at all. So yes, these are a few things that I wanted to tell you, and that's all. Wishing you the best. If you have any queries, by the way, can you do a favor? Can you tell me when is your biology FA1 examination in the comment section? Because I really want to know. I am planning something. If at all I could execute that, it will help you out. Yes, so that's all. Lots of love. Bye bye. Do not forget to hit the subscribe button.